our channel. This is Science Lectures. Please, thanks for being with us. Consider subscribing and like our videos and comment in the comment section. Please. We are here to solve the science mystery, indeed. So today we are going to look at reproduction in bacteria. We are still continuing with our series of Monera, Kingdom Monera, where we said we have basically bacteria and cyanobacteria. So today we are going to look at reproduction in bacteria. We saw classification, how we classify bacteria. We saw a general overview of the structure of the bacterial cell and how it does whatever and which functions are performed by each of them. So today we are going to look at reproduction in bacteria. And we know that reproduction is either sexual. Reproduction is always either sexual or asexual. Asexual. So of course sexual involves the use of gametes. We'll have gametes in asexual, in sexual, then asexual, of course, no gamete use. So reproduction, how does it happen asexually in bacteria? So asexually in bacteria, it happens by a process known as binary fission. Binary fission. Fission means splitting, binary means two. So in binary fission, we'll have our bacterial cell, let's say bacterial cell A, then it splits into two bacterial cells. Let's say it's producing bacterial cell A and bacterial cell A. So that is binary fission, where we have formation of two daughter cells from one bacterial cell simply by dividing. However, before this division happens, we'll have what we call doubling. During this division, before the real division happening, we'll have doubling of the cellular contents have doubling of the cellular contents of the parent cell. So after doubling of the cellular contents, then the cell can divide into two daughter cells that are the bacterial cells from this binary fission. So for such a process to happen, you'll find that bacteria can do this very rapidly. So bacteria are capable of dividing rapidly, and this is one of the characteristics that makes them uneasy to be controlled. Rapid cellular divisions can happen, and we can find that some bacteria can even divide into half for every 20 to 30 minutes. Every 20 to 30 minutes. This is even less than an hour, dividing. So in the first round, let's say we are having one bacteria yielding 2. These 2 can become 4. The 4 can become later like around 8. The 4 can even become 16, because it's 2 to the power n, the rate of division. So they are rapid multiple cell divisions that can happen in the shortest period of time, which is 20 to 30 minutes. So this is a good characteristic for the survival and multiplication of bacteria. However, for bacteria to divide like this, there are certain requirements that will be present in the environment. So what are some of these requirements? Let's say the bacteria having nutrients, presence of the required nutrients for growth. We know that bacteria require macro and micronutrients, and some require heavy metals. Macronutrients like sulfur, phosphorus, potassium, magnesium, hydrogen, oxygen, carbon. Then micronutrients like selenium, like tungsten, vanadium, cobalt, silicon, all those are required. Then heavy metals, some will require heavy metals to work as core factors for their enzymes. So that is nutrients being present. Let's say we are having the food present, we have the water there, then we have no congestion and others. But what are some of the things that will limit growth of these bacteria in the environment? So what limits growth? This rapid multiplication of the bacteria. Number one is limited food. They don't have food, they don't have water, they lack the nutrients. The pH is unfavorable for them. Let's say because all bacteria have specific pH which they thrive. Some like a very high pH, some like moderate pH, others. So those ones that require very high pH are alkalophiles. Alkalophiles will require very high pHs of around 10 to 11. That pH of around 10 to 11. Then acidophiles acidophiles will require pHs even below 2, below pH of 2. Then most bacteria will enjoy a pH of around 5 to 9, pH values of around 5 to 9. So pH is also a factor that can affect bacterial growth. pH, we have food, water, nutrients. Then which other thing can affect? Accumulation of wastes. Accumulation of wastes. So when wastes accumulate, some bacteria will tend to die off. So that means it is limiting the multiplication of this bacteria. Then what else can reduce on the number of our bacteria? Presence of toxins. Such a toxin can be an antibiotic. Let's say we have a penicillin next to gram-positive bacteria. It will cause their death. So presence of penicillins. Which other factor can limit presence of this? The bacteria. Predators, presence of the predators. For example, the protozoans. Some protozoans feed on bacteria as a source of food. So if we have protozoan feeding on this bacteria, engulfing them, they engulf them and digest them using their enzymes. This will limit the growth of the bacteria. However, when bacteria undergo harsh conditions, if we have bacteria facing harsh conditions, what are some of these harsh conditions? Like increase in UV light. We have high UV light radiations. Then environments that have limited water. If we have limited water in the environment, these bacteria, under the influence of such harsh conditions, they will develop what we call endospores. So this endospore formation, when they form the endospores will help them survive in the harsh conditions. And some endospores are too dangerous that they are even resistant to high temperatures and chemicals. For example, the endospores of the bacteria Bacillus anthracis, the one that causes anthrax. So basically, that is what is involved there. Some of the hints we need to know when we are doing this reproduction in bacteria. So basically, this was sexual reproduction in bacteria, where we have binary fission basically taking place. However, we also need to look at asexual reproduction in bacteria, asexual reproduction, and in our sexual reproduction, which was basically binary fission, we have the bacteria being formed exactly similar to the mother cell. But in asexual reproduction, we'll have some genetic recombinations. And asexual reproduction is divided into three basic forms of reproduction. Number one is conjugation. We are going to have a look at conjugation. Another form of asexual reproduction is transformation. And then finally, we have another form called transduction. Transduction is another form of asexual reproduction. So we are going to start off with conjugation. What is conjugation? So in conjugation, you remember a structure. There is a structure we call the pilus on the bacterial cell. The pilus. So the pilus will enable conjugation to take place and presence of another structure known as the plasmid. So in conjugation, we are doing recombination of DNA. We basically have recombination of DNA by exchange of the plasmid, strands. So one of the strands of the plasmid has two strands basically, but will transfer one plasmid from bacterial cell A to another cell. Let's imagine this is our bacterial cell and it has a chromosomal DNA. It has its DNA. Let's say this is its DNA with a single chromosome. Then plasmid, let's imagine this is our plasmid. So this is the plasmid, and this is the chromosome or the DNA, the actual DNA of the bacterial cell. Then we have another bacterial cell, also having this one simply having just the chromosomal DNA. So this one that carries the plasmid, we call it the F positive cell, because it is fertility positive. It is carrying the plasmid. Then this one that does not possess the plasmid is called the F minus cell, because it doesn't have the plasmid. The F positive is the fertility positive, the one possessing the plasmid. The F negative is the fertility negative that doesn't possess the plasmid but has the actual bacterial DNA. So what will happen is that the pili will develop. This is the pilus. Our pilus will develop. And the aim of this pilus is this. The pilus, if we are having our cell, bacterial cell, and we have this bacterial cell, we say it has the DNA, but it has the plasmid. Then we say this one has only the DNA. However, we say a pilus is formed somewhere there. So the pilus does this. The plasmid will transfer one of its plasmid strands to the F minus cell, the F negative cell, the fertility negative, from the fertility positive cell. Then this plasmid will be received by the F negative cell. And when the plasmid is received, this is what we have currently. At that point, we have the DNA there, this one possessing only one strand of the plasmid. However, this other cell, this is our DNA, has gotten a plasmid strand there. So the final step, finally, what will happen is that there will be formation. The plasmid that was, the cell that was F positive, will use the remaining strand to form a complementary strand to recover back its plasmid with two strands. Then also the one that has received one strand will synthesize a complementary strand, will also develop a complementary strand to also recover a double-stranded plasmid. So that is how conjugation will help, and this process is very important in that it will cause recombination. We talked about recombination, so we are altering the genetic. So plasmids are plasmids are very important in antibiotic resistance, in the bacterial cells, antibiotic resistance. So if the F positive cell had a plasmid that had a gene for antibiotic resistance, it will give the gene to the F negative cell, hence very important for the survival of cells. Even not only antibiotic resistance, but other important genes that the cells might require. So that is it with conjugation as one form of sexual reproduction. So transformation is where we have DNA fragments from cells, from other bacterial cells. Let's say these bacterial cells are dead, they die, or they fragment, they burst. Fragmentation of bacterial cells within the environment. So this fragmentation of the cells will release the genome, the DNA. So if we are having, this is the cell containing genes like capital A, capital B, Okay, the genome, let's say this is the genome in large amounts, capital A, capital B, capital A, capital B. And these cell fragments, the cell bursts or fragments or bursts. When this cell bursts, another bacterial cell, this is bacterial cell A, then bacterial cell B is around here. Let's say it is having gene small a and gene small b. Then when it bursts, when this one bursts, we expect it to release this gene a small b. Let's say here it had also a small b, a small b. In fact, yeah, a small b, then a small b. That is what it is having. But when it bursts, let's say it releases, this one bursts now, releases its capital A, capital B into the environment. This other cell takes up the capital A and capital B, or capital A only. Then finally it becomes capital A, small b, or small a, capital B. So let's say it becomes like this. This is what we call transformation. We are having DNA fragments in the environment or DNA fragments from other bacteria that bursts or fragment, releasing this genome that is later incorporated into other bacterial cells. So this one also leads to what we call recombination. So we have a variation from the cell that we initially had, the genome of the cell that we initially had, to the genome of the cell we currently have. So that is it with transformation. Then let's dive into transduction and see what happens in transduction. So we have transduction. So in transduction, we'll use what we call bacteriophages. The key components here are bacteriophages. So what will happen in transduction is that the bacteriophage will pick DNA, will pick the genetic material or DNA from a specific bacteria, then it puts that DNA or genetic material into another bacteria. So that is basically what transduction is all about. So let's say we are having, this is our bacteriophage, then this bacteriophage, then it comes to the bacterial cell, this is the bacterial cell, of course having some DNA there. Then when this bacteriophage comes, it will use the host machinery, host cells machinery to manufacture other bacteriophages containing both the host DNA, but in this process, its DNA, the DNA of the bacteriophage will then contain the host DNA at the end of it all, the host DNA. So we are having a bacteriophage attaching onto this bacteria. Then after attaching onto the bacteria, it will use the host cell machinery. It uses the host cell machinery. I say that is our host cell. It will use its machinery to manufacture many bacteriophages. However, when these bacteriophages form, at the end of it all, some bacteriophages will have the DNA of the bacteriophage together with the DNA of the host cell. So that is what will happen in the machinery. Then after this happening in the using the host cell machinery to manufacture the DNA, this bacteriophage that is infected that is carrying both genes will go and infect other bacterial cells let's say we are having, this is another bacterial cell. Then this guy comes in with the DNA, the bacteriophage DNA inside there zoomed as blue and having some black. Then let's say that this bacteria is also having its DNA inside there that is red. So what will happen when the bacteriophage brings in this DNA, the one which is both blue and black, blue, then the black also present there. This genome coming from the bacteriophage has the blue. Let's imagine it has the blue, then the black. When it is fixed into this bacterial cell, the new bacterial cell now we are having, it will be, of course, it is blue and black. But remember, this black comes from a bacterial cell. The blue, sorry, comes from the bacterial cell we initially saw, the bacteriophage infecting. So at the end of it all, we'll have this, the genome carried from the bacterial cell that was this side to into the genome of our new cell. So finally, the genome will become we'll be with the red. We'll have a red color, and then we'll as well have the blue color, the blue on it. We'll as well have the blue on it. So that is what we call transduction. We are getting genetic material from a different bacteria, but we are fixing it into the genome of another bacteria. So this is very important in things like biotechnology. In biotechnology, where they need to get certain genes and incorporate them into a specific bacteria to effect certain processes. So basically, this is reproduction in bacterial cells. We saw sexual reproduction where we have binary fission. We saw asexual reproduction where we have binary fission. Then we've seen also this type of division that brings about recombination, which involves conjugation, where we simply have a plasmid F positive giving the F negative. Then we saw transformation, where we are having DNA fragments incorporated into our DNA. Then transduction, where bacteriophages pick DNA from a specific bacteria, then incorporate them into other bacteria. So that is it for reproduction in bacteria. Thanks for watching, please.